We open with the customary cautionary slide. Basically, we're stressing that you do not want to use these podcasts or these lab test reviews as a substitute for either going to biosis or attending lab and performing the exercises as detailed in your lab manual. Um, if you fail to do um, either of those activities, you're going to um, not do as well in the practical. And of course, we don't want that to happen. This is simply meant to augment your ability to successfully navigate the practical when it comes about. So we open with organic molecules, and in this lab, what we're doing is we're representing some biologically relevant molecules using ball and stick models. And what we're showing you here are which balls indicate which atoms, and the sticks indicate covalent bonds. So note that the hydrogens are white, the carbons are black, the nitrogens are light blue, the oxygens are red, and the phosphorus atoms are purple. So let's keep that in mind as we navigate our way through the rest of these compounds. Now, what we want to do here is we want to identify each of these molecules by name. So let's start out with the upper left-hand corner. This is an oxygen molecule. Notice that it's held together by a double covalent bond. Now, first thing we want to understand is what is a covalent bond. A covalent bond is one of two types of chemical bonds that exist between atoms in the same molecule, the other being an ionic bond. In a covalent bond, what happens is that a pair of electrons is shared between two atoms. So each one of these sticks here represents a pair of electrons that spend some time around atom number 1 and some time around atom number 2. One of the things that we want to understand about the formation of chemical bonds is what governs their formation. What is it that dictates how many chemical bonds a particular element can form? And what you have to understand in order to have that make sense is you have to have some knowledge of the periodic table. So what would be a good idea for you to do if you have the opportunity? For those in my section, in the lecture on chemistry that we had in class, there was a slide that had a very good periodic table in one of the PDFs that I posted up on Angel. You can also Google a periodic table and print it out. It's not a bad idea. And with that in front of you, we can have a brief discussion about how it is that atoms make and break chemical bonds. So one of the things you have to understand about making and breaking chemical bonds is that chemical bonds exist between the outer shell of electrons of any element. Basically, remember that an atom consists of a nucleus that contains protons and neutrons, and then electrons which orbit the nucleus at discrete distances, and these discrete distances are what we call shells, basically like floors in a hotel. And the way that a lot of atoms are set up, the atoms that we're going to deal with when we talk about the chemistry of life, biochemistry, is that the first shell that orbits the nucleus of an atom can hold a maximum of 2 electrons. After that, there's no way to load any more electrons into that shell. We have to move up to the next shell, which can host a maximum of 8 electrons. The third shell can host a maximum of 8 electrons as well. And then as we get up into the heavier elements, we start to be able to accommodate more than 8 electrons. But the chemistry of those elements is going to come up very seldom in an A&P course or in a biochemistry course. So we'll just concern ourselves with what we call the octet rule, which is essentially the tendency for any element to try and get a full outer shell of electrons. For the light elements, hydrogen and helium, that full shell is represented by 2 electrons, and for heavier elements, that full shell is represented by 8 electrons. A covalent bond represents the sharing of a pair of electrons between 2 elements. So the way to determine the number of covalent bonds that an element can form is to look at the outer shell, count the number of electrons in it, and then subtract that number from 8. And that'll tell you the number of covalent bonds that element can form. So if you've got your periodic table in front of you and you're looking here, let's take oxygen. Okay, oxygen has 6 electrons in its outer shell. It has a total of 8 electrons. Those 6 electrons in the outer shell are the valence electrons. If we subtract 6 from 8, we get the number 2, and that's the number of covalent bonds that oxygen can form with another element. Okay, in this case, oxygen forms 2 covalent bonds with another oxygen molecule, and that results in a full outer shell for both oxygen atoms. And as a result, that's what we call a stable compound. When that outer shell is full, that species, whether it's an ion or an atom, is in a low energy state, and that satisfies the second law of thermodynamics, which basically states that whenever possible, components in a system try and find the low energy state as quickly as possible, because that's the easy thing to do. Okay, it doesn't take an energy input in order to maintain. So as we move around these compounds, we'll sort of refer to the periodic table and the number of covalent bonds that can be formed, and this will start to make more sense to you. Down here, B, okay, this was an oxygen molecule, O2. B down here, this is carbon dioxide. Okay, now carbon has 4 electrons in its outer shell. If you look on the periodic table, it's in group 4A. And so 8 minus 4 is 4, so carbon can form 4 covalent bonds, and that's exactly what you see here, 1, 2, 3, 4. And remember what we said about oxygen. Oxygen can form 2 covalent bonds, and that's exactly what you see for each oxygen molecule, 1, 2 for this one, and 1, 2 for this one. And so as a result of having full outer shells in all 3 atoms in this molecule, you have a stable compound, and that's essentially the kind of product you're going to get in a chemical reaction. You're going to try and produce a stable compound. This molecule here is called methane. The structural formula is CH4, a central carbon bound to 4 other hydrogen atoms. Now remember, hydrogen has a max occupancy in its outer shell of 2 electrons, while carbon has a max occupancy of 8. So let's see if we satisfy the stability requirements for each of these atoms in methane. Well, let's look at the hydrogens first. 2 electrons is represented by this stick, stable atom. 2 electrons here, stable atom. 2 electrons here, stable atom. There's 2 electrons here. You can't see the stick there because it's hidden by this hydrogen atom. So this is a stable hydrogen. How about carbon? It should have 8 electrons orbiting its outer shell in order to be stable. So let's count: 2, 4, 6, 8. This is a stable molecule. The carbon is stable. The hydrogens are stable. Okay. Of these, the only one that's considered to be organic is methane. The definition of an organic molecule is a molecule that contains a carbon that's bound to at least 1 hydrogen. That's why carbon dioxide does not qualify as an organic molecule, even though it contains carbon. Now, a question that you might want to ask is, okay, anyway, let's answer each of these questions here. Oxygen, right? The oxygen molecule, carbon dioxide, water over here, right? An oxygen atom that can form 2 covalent bonds, and a hydrogen atom that can form 1, okay? Stable hydrogen, stable oxygen, 2, 4, and then the remaining 4 electrons aren't really pictured here because we can't represent them in this ball and stick model, but they would be basically on the in the outer shell of the oxygen atom. So the oxygen here is a stable molecule. Okay. So this is water, and this, of course, is methane. Okay. So which of these molecules is considered an organic molecule? That's D. What kind of covalent bonds are seen in A and B and are not present in C and D? And the answer is double covalent bonds. Now, before we move on to the rest of the presentation, I want to talk a little bit about why carbon is so critical as a basis for generating the molecules that we think of as vital to living systems. And there's one major reason that carbon is the basis of most molecules that we think of as vital to life processes, and that's its ability to form long chains and to form rings with itself. And what that does is it provides a vast array of potential structures that you can build using carbon as a basis. Okay, so that's why carbon is the basis of life as we know it. The other elements in the periodic table can't really do that nearly as effectively, and as a result, they don't have the potential for the complexity of molecules that have to perform life functions. Things like proteins and nucleic acids, fatty acids, and so forth. Okay, so this is why life is based on carbon. So this is the covalent bond. Now, remember I told you that there's another major kind of chemical bond that we want to be aware of, and that's the ionic bond. And in the ionic bond, what happens is that instead of sharing electrons between two atoms in a molecule, an electron is actually handed over completely from one atom to another, and what holds the two atoms together is their opposite charge and their electrostatic attraction for each other. You see that in compounds like sodium chloride, for instance. If you have your periodic table in front of you and you look up sodium, what you'll find is that it has one electron in its outer shell, and in the shell just underneath that, there's 8 electrons. And so the tendency for sodium when it tries to form compounds with other elements is to give up that outer electron, because that's the easiest thing to do, and to form a stable, positively charged atom called a cation. Now, where does that electron go? Well, that electron goes to the other participant in the compound. Now, let's say that the other participant is a chlorine atom. Well, chlorine is a group 7A atom, so it has 7 electrons in its outer shell, which means that it can host one more. So chlorine will pick up that electron from sodium, and it'll actually form a negatively charged atom called an anion. And what will hold the sodium and the chloride together is their electrostatic attraction. Okay? There's a third type of interaction that we want to talk about while we're looking at this molecule of water here, and that's a type of molecular interaction that's called hydrogen bonding. So if we were to draw another water molecule, okay, so let's for fun just sketch another water molecule right next to this one. So this will be our oxygen, okay, and then we'll draw a couple of hydrogens attached to it. So there's our hydrogen here. This is our covalent bond, and then we'll draw our atoms. So there's one hydrogen atom, and out here there's another one that's kind of tough to see, but take my word for it, it's there. One of the things that you'll notice in some covalent compounds is that this sharing of atoms between, or the sharing of electrons between two different atoms, isn't necessarily equal. There's a property of certain elements in the periodic table called electronegativity. Now, the elements that have this property tend to be in the upper right-hand corner of the periodic table between the group 8A elements and the group 4A elements. So basically everything to the right of carbon in the periodic table in the first, call it 3 rows. Okay, so atoms like oxygen, nitrogen, phosphorus, sulfur, okay, are all examples of atoms that have this electronegativity property. And what that means is if they participate in a covalent bond with an atom that's either in the same column as carbon or to the left of carbon, the sharing of electrons won't be equal. And so what you'll have is a slight separation of charge. And the way we indicate that when we look at a molecule of water is by using the Greek letter delta, okay? And then on the oxygen we put a slight negative charge because it tends to pull electrons towards it. While down here by the hydrogen we would put a slight positive charge, okay? And that would be on each hydrogen. All right? Now what this means then is that for every water molecule you've got this same characteristic. So let's put it for this water molecule as well, okay? There's our hydrogen out here, okay, and down here. Okay, now what this means is that when two water molecules approach each other, they're going to try and satisfy that second law of thermodynamics again, which is to arrange themselves in the lowest energy arrangement that they can think of. Okay, and the lowest energy arrangement is to get the pluses near the minuses and the minuses near the pluses, and this results in a sort of an electrostatic attraction between two parts of two different molecules. The slightly negatively charged oxygen of one water molecule and the slightly positively charged hydrogen of another water molecule. This is a bond called a hydrogen bond. Okay, it's really more of an electrostatic attraction than anything else. And it accounts for a lot of different properties of water, such as water's ability to interact with itself. This produces the phenomenon of surface tension. If you've ever seen those insects that can walk across the surface of water like Jesus did, they use surface tension to do that. Surface tension is the attraction of water more for itself than for the air above it, and this results in enough upward force to support the weight of the insect. It also accounts for the tendency of water to expand when it freezes. When a compound or an element goes from a liquid to a solid, what happens is the amount of molecular motion slows down. Okay, in a solid, the components of the solid sit at fixed points and vibrate. There's very little molecular motion. That's why solids tend to retain their shape, regardless of the container that they're in. In a liquid, there's movement of molecules past each other. They have a little bit of kinetic energy, and as a result, liquids will form to the shape of their container because of this ability to kind of move around in that particular phase. And then a gas has the most kinetic energy, and as a result, a gas will fill all parts of a container. That's why gases generally have to be stored in metal cylinders, okay, so that you don't lose the gas. So in a water molecule, this interaction is primarily between molecules that have polarity. Okay, we say that this is a polar covalent bond because of the unequal sharing of electrons and the resulting separation of charge. You also see this hydrogen bonding when we look at the effect of capillary action. If you've ever donated blood and they take one of those capillary tubes, touch it to the drop of blood on your finger, you'll see that the blood will wick up the tube all the way up to the indicator mark. Well, why does that happen? Because of the attraction of the polar water for the polar glass. Okay, the glass is primarily silicon dioxide, and remember that polar things like to associate with polar things, as we discussed in lecture, and the reason for that is that that's a low energy situation. Okay, this interaction generally is again an interdigitating of charge, the negatives being near the positives being near the negatives, and as a result, that's a low energy state. And so what happens as the water in the blood in the capillary tube is attracted to the walls of the tube, it draws other water molecules with it, and the water will wick up the tube. This is also why water can get to the tops of tall trees, right? Because the water is attracted to the cellulose, the wood in the tree, because that also is a polar molecule. It's made up primarily of sugars. And then as the water evaporates from the leaves, um, that produces enough draw to go from the roots all the way to the shoots, regardless of the tree's height. This also accounts for the reason that ice floats. Okay, um, when water transitions from a liquid to a solid, the movement of the water molecules past each other has to stop. And when it does, those water molecules have to be oriented in a way like I've drawn here, okay, between these two water molecules. And what that does is that puts empty space between the water molecules. As a result, ice is less dense than liquid water. That's why ice floats in water. Y'all know this intuitively, but this is the chemical explanation for it. Ice also um, takes up more volume than the equivalent volume of liquid water. This is why if you leave a can of juice in the car on a cold night, you'll come out the next day and the can will have split open, and that's because the water went from a liquid to a solid. When it froze, it expanded in volume, it split the can open. Okay, so all this you've seen, you just maybe didn't know why these things were happening. Okay, so maybe that helps these interactions make a little bit more sense. So where do we see hydrogen bonding? We see hydrogen bonding between polar molecules that contain hydrogen atoms bound to electronegative atoms. And so obviously this exists in water, but it'll also exist in a variety of other compounds that we'll come across. Okay, here we have a little matching activity. There's the answers. Okay, we actually just went through them. Here's another matching activity. This is a matching activity for functional groups, and what we want to know is which functional group is which. Now, what the heck is a functional group? It's a cluster of atoms. It's attached to a larger molecule. Okay, and the reason we make a big deal out of functional groups is because they can impart unique chemical properties to the molecule that they're attached to. Let's take each of these functional groups one at a time. Okay, this is a methyl group, CH3. This covalent bond would be attached to another molecule, hence the carbon would have 4 bonds, each to a separate atom. Why are methyl groups a big deal? Methyl groups are a big deal, number one, because they contain carbon-hydrogen bonds. And one of the things we're going to discover as we move through this presentation is that the carbon-hydrogen bond is significant because it contains more chemical potential energy than any other bond that a living system is going to encounter. And when these bonds are split, when they're broken down, energy can be harvested and used to make the energy currency for the body, which, as my class knows, is ATP, adenosine triphosphate. That's the source of energy that molecules in the body can immediately use to do work. It's kind of a quirk of fate that you can't use the chemical potential energy that's locked up in the chemical bonds of the food you eat directly to do work. You have to transfer that energy to a molecule of ATP, and then you can immediately use that to perform life processes. It's similar to the way that the engine in your car works, right? You know that the engine in your car understands one kind of fuel, okay, gasoline. And what is gasoline? Gasoline is made up primarily of an 8-carbon molecule called octane. Okay, what happens with octane in your tank is that it gets to the carburetor mixes the gasoline with air, the air contains oxygen. The spark plug, when it comes into contact with this oxygen gas mixture, will shoot a spark. That spark will cause the gasoline to explode, to oxidize. The octane will be reduced to carbon dioxide and water, and the other products that will be produced will be pressure, light, and heat. Okay, and what that explosion does is it moves pistons inside your engine, so that produces kinetic energy. That's going to turn a crankshaft, and that's eventually going to turn the axle around which the wheels of your car revolve. Okay, so that's an example of energy transformation. But where does gasoline come from? It comes from crude oil. Well, if you've ever seen crude oil, it's a thick black sludge that comes out of the ground. If you were to put crude oil in your gas tank, your engine wouldn't understand it. Now, it doesn't mean there's no energy there. It just means that your engine isn't set up to use that energy to produce movement. And the same thing is true in your body. There's all kinds of chemical potential energy in carbohydrates and fats and in proteins, but the cell can't use those directly to do work. They have to extract the energy from the chemical bonds in those molecules and transfer that energy to ATP so that we can do work. So there's your methyl group. Methyl groups are also important because they're used to modify molecules like DNA. They're used to modify proteins, um, and when they're used in this way, they often act as signals. Okay, and that's important in processes such as gene regulation. For instance, there's certain times when certain information in the DNA needs to be used by cells, and other times when we don't want to use it. One of the ways that we can shut that information off is by methylating DNA. Okay, there's a lot of other uses for methyl groups, and we'll see those as we kind of move around this presentation. This is a hydroxyl group. Remember, oxygen can form two covalent bonds. One here is with a hydrogen, and the other is with another molecule. Now, you generally find hydroxyl groups attached to alcohols. Okay, another place that you'll find hydroxyl groups is on steroids. Okay, the significance of hydroxyl groups is that they can chemically react with other functional groups to form novel covalent bonds. For instance, hydroxyl groups can very easily interact with carboxyl groups, okay, to form what we would call an ester, okay. So if I were to draw this, I'll draw this using a biochemical shorthand here. Let's just kind of whip this up. This would be our carboxyl group, okay. All right. And then there would be some molecule hooked to this thing. This could be coming off of an amino acid, or it could be coming off a fatty acid. We'll just put an R here to represent the rest of the molecule. And then this could be our hydroxyl group, okay? There's our hydroxyl group, our OH, and then it would be connected to some other molecule, okay? And then what happens is that generally, this is in living systems an enzyme catalyzed reaction, and what happens is that a novel bond is formed between this atom and this atom, okay? And when that occurs, the product that you make, we'll draw it down here, has this kind of a formula, R, C double bond O, okay? And then another O, and then another R, okay? An organic chemist would call that an ester. And what we lose in between these two molecules is we lose the components of a water molecule. Okay, so this is what we call a dehydration synthesis. So a water molecule would leave, okay, and we would form this novel linkage between these two groups. Okay, so that's one of the reasons that hydroxyl groups are important. Whenever you find a hydroxyl group on something, that's likely to make it polar. Okay, because remember, the oxygen is electronegative. It tends to pull electrons towards it, and that tends to generate a separation of charge. Okay, so those are a couple of the interesting aspects of hydroxyl groups. This down here, this is an amino group. This is a nitrogen. Nitrogen has 5 electrons in its outer shell. 8 minus 5 is 3, so it can form 3 covalent bonds. There's 1, there's 2, and there's your third covalent bond. Also remember, right, that accounts for 6 total electrons right there, which means nitrogen always has a lone pair of electrons in its outer shell that's available to interact with, for instance, charged species, okay, like protons, for instance. And that's why amino groups are generally what we call basic groups. Okay, basic groups are those that can scavenge protons from the surrounding solution, cause the pH to go up. Okay, so that's one of the significances of an amino group. You also find amino groups on amino acids. Okay, an amino acid will have both an amino group and a carboxylic acid group, which is what this is, on the same molecule. Okay, all right. Um, Other places that you find amino groups, you find them in nucleotides. Okay, you find them in amino acids. You can sometimes find them in phospholipids. Um, and again, they impart polarity to the molecule that they're attached to, because the nitrogen is also an electronegative atom. Okay. All right, the carboxylic acid group here. Okay. Now this is a carboxylic acid group. I already drew it up here for you. The reason that this thing is called a carboxylic acid is because it will lose this molecule, this atom here, which is a hydrogen atom, but that hydrogen atom, when it leaves, generally leaves its electrons behind on the oxygen. And so when you draw this ionization here, okay, let's draw this carboxylic acid. We'll put an R here. We'll put a carbon there. We'll put a double bond there. Okay, and we'll put the OH here. Okay, and what happens when we ionize is we lose the proton that goes off into the solution. The solution is a aqueous or water solution. Okay, there's our positive charge, and then we form this guy. As a negative charge. And so what happens as a result of this ionization is that the pH of the solution that the carboxylic acid group is in begins to drop. Okay? Now, why does this happen? Why does this group give up its proton so easily? Well, the reason for that is that the negatively charged molecule that's left behind is particularly stable because it can take this negative charge and it can spread it out over 3 atoms. And the way that happens is that these electrons can hop down here, okay, and these electrons can hop up here, all right, and you can form now an alternate structure that looks like this. And so what that means is that this negatively charged molecule is more stable. The more atoms that you can spread the charge out around, the more stable it's going to be. Okay, so that's just a way to kind of clue you in as to why this thing coughs up its protons. Okay, so that's a carboxylic acid group. Obviously, it's a polar group. The carbon here happens to have a partial positive charge. The oxygens have a partial negative charge. Okay, so this is one of the reasons why it can form new chemical bonds with hydroxyl groups that happen to have a lot of electrons around them. Okay, in organic chemistry, that's what's called ester formation. So that's the mechanics of why these groups are important. You find this cluster on fatty acids. You also find it on amino acids. Okay, and this is our matching here: the methyl group, the amino, the hydroxyl, and the carboxyl. Okay, so now we can move over here. Which of the 2 above functional groups are components of all amino acids? Well, there you go, D and C. Which one of the above functional groups is found in the glycerol molecule? And that's B, and you actually find 3 of those in the glycerol molecule. It's a triol, okay, an alcohol with 3 hydroxyls. And the next question is functional group C a component of fatty acids? And the answer is absolutely it is, okay? And so is functional group D. Okay, moving on. What kind of organic molecule is this? This is an amino acid. Okay, why is it called an amino acid? Well, because it has an acid group and an amino group. Okay, the acid group is this guy right here, the carboxylic acid cluster. C double bond O, OH. Okay, and the amino group is here on the back end. Okay, that's the amino group. This is called the alpha carbon. Now, the alpha carbon in an amino acid is bound to a hydrogen. Okay, so this part that I'm circling here in blue, all amino acids have this. And then the, the last part of the amino acid, the part that I'm circling here in green, is what we call the R group. Now, the R group can be any of 20 different collections of atoms that are bound to the alpha carbon. And depending on the cluster of atoms that's bound to the alpha carbon, the amino acid can be classified either as charged, as polar, or as nonpolar. And so here we see what it is about the building blocks of amino acids that allow us to make so many different molecular tools using just 20 building blocks. Because we can hook these amino acids together in any way we see fit, and at any length and in any order, the number of potential molecular tools we can build is almost infinite. Now, how is it that living systems know exactly what tools to build in order to carry out life functions? And the answer is evolution and natural selection have created information in the DNA, which is then copied to messenger RNA that is transported out of the nucleus, and those instructions are read by ribosomes, and those instructions dictate which amino acids to incorporate into a particular protein in a precise order, and how much of that protein to make. And that protein will have a specific job to do inside the cell. It might be a membrane receptor, it might be a channel, a molecule channel in the membrane, it might be an enzyme, it could be any of a number of different molecular tools. And it's only through millions of years of evolution that we've come up with the information necessary to build these perfect tools in the perfect way. We want to now identify this organic molecule. This is a tripeptide. How many peptide bonds are in this molecule? The answer is 2. And is the indicated bond a peptide bond? And the answer there is no. Now, first thing we want to do is describe exactly what we mean by a peptide bond. So let's circle the peptide bonds in this molecule, okay? There's one, okay? And there's another one. So in a tripeptide, you've got 2 peptide bonds. This peptide bond is a type of covalent bond, okay? If I were to draw the chemical representation of a peptide bond, it would be a covalent bond between what an organic chemist would call a carbonyl carbon, okay, and the amino group of another atom. Okay, so there we go with that. Okay. Okay, this would be hooked to the rest of the amino acid of amino acid number 1, and this will be hooked to the rest of the amino acid of amino acid number 2. Okay, so the peptide bond, we're going to circle again in yellow, is this guy right here. Okay, he only exists between these 2 particular types of atoms. This is part of the covalent backbone that holds proteins together. The nice thing about these proteins is as you assemble them one after the other, you generate what's called the primary structure by forming these peptide bonds one at a time. As you finish building the protein, what's going to happen next is that depending on where the R groups are in the long protein, that protein will begin to fold up in such a way as the nonpolar R groups in the amino acid residues will find the other nonpolar R groups. The polar groups, R groups in the amino acids in the chain will find other polar R groups or will associate with water. And the charged R groups will find R groups of the opposite charge. And that, along with hydrogen bonding, goes a long way towards producing a three-dimensional shape that's perfect for doing a particular job inside the cell. Now, if we wanted to box out each of these different amino acids in this tripeptide, let's show you. This would be one amino acid right here. Okay. The next amino acid, we use a different color, would be this guy right here. And the last amino acid would be this guy right here. Okay. And this is what we call tripeptide because it's got 3 amino acid residues. Now it turns out this particular tripeptide is made up completely of glycine. Okay, the R group for the amino acid glycine is simply a hydrogen atom, as indicated here and here, okay, and there. But we could have put any kind of tripeptide together. We could have used alanine, which has a methyl group as its R group, or we could have used isoleucine, which has a longer carbohydrate chain as its R group, or we could have used aspartate, which has a negatively charged R group. Doesn't matter, okay? What dictates the order in which we hook the amino acids together is the information that's contained in the gene, and the gene is the result of a lot of tinkering on the part of Mother Nature to come up with a protein that provides an advantage for the organism that expresses it. Okay? All right. What kind of organic molecule is this? This is a steroid. Now, what's the significance of steroids? Well, they form the backbone of steroid hormones such as testosterone and estrogen. They also are part of a molecule called cholesterol, which has a lot of different roles. Cholesterol is found in bile salts, where it helps emulsify the fats in the food that we eat. It's also an important component in controlling the fluidity of cell membranes, and it's also basically a structural component of vehicles that transport lipids around in the lymphatic system and in the circulatory system, which are called lipoproteins. Okay, they're carriers of hydrophobic molecules, and a hydrophobic molecule is one that is nonpolar. And as you can see, a steroid is a type of lipid because it is basically nonpolar. If you look at this fused ring system, what you find is that it's basically made up of carbon and hydrogen. As a result, it's rather greasy. Okay, likes to associate with other greasy molecules like oils and fats and so forth. There is an electronegative atom on here. There's an oxygen there, but compared to the rest of the molecule, that's not going to really change its hydrophobic characteristics very much. So again, lipids like to associate with other lipids, and that's why you tend to see the steroid molecule generally embedded in things like triglycerides, cell membranes, um, or or fat in the digestive system. Okay, so important molecule here. It has a notice that all these carbon-containing molecules have a three-dimensional shape, and the reason for that is that carbon can bind a maximum of 4 other atoms. Doesn't really matter what the identity of those atoms are, but in order to minimize the repulsion between the atoms that carbon's bound to, when carbon is bound to 4 other atoms, it takes on what we call a tetrahedral shape, which is kind of like a TP with an antenna on top. when carbon is bound to two other atoms, it forms a straight line. That's called linear. And when carbon is only bound to three other atoms, it's flat like a pancake. Okay, so here's an example of carbon bound to three other atoms. 1, 2, and 3. There's a double bond here. Okay. Now, why is that a big deal? Well, because shape goes a long way towards establishing the function of organic molecules in living systems. So we want to be aware of the fact that these molecules have a three-dimensional shape and not just a flat shape like we might imagine if we looked at them on a piece of paper drawn out. Okay, consider the ratio of the three elements that are seen in this molecule. What about this ratio would suggest that this molecule is a carb and not a lipid? And the answer is the hydrogen to oxygen ratio, which is approximately 2 to 1. Okay, this is a carbohydrate. This is a disaccharide. This is sucrose. Okay, and this molecule, what we do is we hook two monosaccharides together, and we do that by forming this novel bond here between the two hydroxyl groups of two adjacent sugars. This bond is called a glycosidic bond, and it's formed by dehydration synthesis. This is a reaction that requires an energy input. It doesn't happen spontaneously, and it's facilitated by enzymes in living systems. Okay, so if this were a lipid, what you'd see is a much greater hydrogen to oxygen ratio, and you'd see far fewer electronegative atoms. Okay, there wouldn't be nearly as many oxygens, and we'll see an example. Well, we saw an example of a lipid when we looked at the steroid nucleus earlier, right? And you could see that the hydrogen to oxygen ratio was huge. Here, it's 2 to 1. Okay, and that immediately tells you this is a carb. Now, what's the significance of carbohydrates? Well, they act as ID tags for cells in the body. They're part of antigens that tell the immune system that certain cells belong to you and that other cells do not. They're also a fuel molecule, particularly in the form of monosaccharides. The carbon-hydrogen bonds in the carbohydrate are again the most energy-intensive bonds. As these bonds are split, the energy is trapped and used to do work. Other places that you find sugars are as structural components of building blocks such as nucleotides. You find sugars in the building blocks of RNA and DNA. You also find them attached to lipids as glycolipids and attached to certain proteins as glycoproteins. Okay, so they can be fuel, they can be signal molecules. They have a lot of different uses. Okay. Next question. Identify this organic molecule. That's glycerol. Okay, this is an alcohol that has three hydroxyl groups, a three-carbon backbone. Okay, this molecule is a component part of triglycerides. Now, what a triglyceride is what happens when you condense three fatty acids onto each of these hydroxyl groups, and you form now an ester bond like we showed you earlier between three distinct fatty acids. Now we'll see what a fatty acid looks like in just a minute, but keep this in mind, okay? Keep this backbone in mind when we get to the slide on triglycerides. Oh, and by the way, this is a polar molecule. Look at all the oxygens here, okay? All right. What molecule is this? Well, this is ATP. This is the universal energy currency in living systems. The element represented by the ball at the tip of the arrow is phosphorus. How many phosphate groups are found in this molecule? And the answer is 3. Now you might say, well, wait a minute now. You said phosphorus and phosphate. What's the difference? Well, a phosphate group is PO4. Okay, so let's draw a circle around a phosphate group. There's one phosphate group. Okay. Here is another phosphate group right there. Okay, they're sharing this oxygen. And here's another phosphate group here. Okay, and they are sharing this oxygen. Okay, so 1, 2, 3 phosphate groups. Okay, How many high energy bonds are found in this molecule? And the answer is 2. Now, you might ask, well, why is that? Well, for reasons that I won't get into right now, phosphate groups have negative charges on them. Okay, they are essentially acids at neutral pH. So they lose protons to the surrounding solution. And so each one of these phosphate groups has a -1 charge. And that means that at neutral pH, these phosphate groups are going to try and repel each other. And so there has to be a lot of energy in the bonds that hold these groups together, or else um, we wouldn't be able to keep these like charges near each other anymore. Okay, and this is why ATP is the universal energy currency. The energy that's stolen from carbohydrates, fats, and proteins is transferred to these high energy bonds, and then this molecule, when these phosphates are split off, releases energy which is used to drive reactions that wouldn't otherwise happen spontaneously. Okay, so you might ask, okay, you talked about high energy bonds. Okay, where are the high energy bonds in this molecule? So let me show you where the high energy bonds are located. We'll do them in yellow. Okay, so let's circle them in yellow. So this is one high energy bond here. Okay, and this is another high energy bond right here. So 3 phosphate groups, 2 high energy bonds, because these guys are pushing off each other so hard. How is this molecule formed? Well, it's formed by a couple of processes. It's a byproduct of glycolysis, which is a metabolic process whereby sugar is split and some of that energy that's released is used to form ATP directly. It's also generated by the Krebs cycle, which happens in the mitochondria matrix of living cells directly. And it's also formed by a process called oxidative phosphorylation, in which highly reduced molecules that have a lot of chemical potential energy in them are slowly stripped of their energy, and that's used ultimately to synthesize ATP from inorganic phosphate, these phosphate groups, and ADP by a membrane protein in the mitochondria called ATP synthase. And you'll learn more about that when we actually get to the metabolism chapter. Okay. Now there's other parts of this molecule that we want to be aware of. We've talked about the phosphates already. This part of the molecule here is called the sugar. Okay, and the sugar in ATP is ribose. It's a sugar with 5 sides. It is a heterocycle. Biologists call it a heterocycle because not every atom in the ring is a carbon. There's an oxygen right there. It's a ribose sugar because both of these groups are hydroxyl groups. In DNA, which is a building block, or I'm sorry, in DNTPs, which are building blocks of DNA, one of these is replaced by simply a hydrogen. Okay, this is actually, in addition to being an energy molecule, it's also a building block for RNA. And then finally out here, this is what's called the nitrogenous base, and in ATP this base is called adenosine. Okay, so there we go. That's the base. Now, it's easy to understand what these guys are. These are where the energy are in the molecule. The ribose is basically a connector between the phosphates and the nitrogenous base. Why do you think this is called a base? Well, if you think back to nitrogen, remember what I told you about nitrogen? Nitrogen has the ability to form 3 covalent bonds. So if we draw a nitrogen molecule, okay, and we draw it bound to 3 other atoms, let's just draw ammonia for fun. Okay, this is ammonia. You've got a lone pair of electrons out here with nothing to do really. And if a proton comes along, and the protons are spontaneously formed in water at a very low rate, this proton can easily interact with these negatively charged electrons, and you can form something called ammonium, NH4 plus. Ignore that. NH4. So there's H. Okay, there's H. There's H. And there's another H. Okay, and this whole thing would have a positive charge. Okay. Now, why is that a base? Well, what does a base do in solution? It steals protons from an aqueous solution, a water-based solution, and that causes the pH to go up. Okay, that's the definition of a base. So this is why this heterocycle here is called a nitrogenous base. It contains nitrogen, and the nitrogen acts in a basic fashion in an aqueous solution. Okay, so that is ATP, the universal energy currency. Next question, what are each of the functional groups? And we already said. Okay, so there's your matching. Next question here, what molecule is this? This is your fatty acid. Now think back to the glycerol molecule that we saw a few slides back. Okay, this is what's going to condense with the glycerol. Okay, is this a saturated molecule? The answer is no, and the reason for that is that there's a double bond in this fatty acid chain. When we say saturated or unsaturated, referring to fatty acids, we mean is every carbon in the fatty acid chain bound to as many hydrogens as it possibly can? And the answer for this fatty acid chain is no. Okay, let's look at each carbon. This carbon's saturated. It's forming 4 bonds, and 2 of them are to hydrogens. This is a saturated carbon. So is this one, this one, this one, this one, and this one. These 2 are not, right? There's a double bond here. They could have potentially bound to hydrogen instead of forming a double bond with each other. The result is this what's called a monounsaturated fatty acid. This carbon is saturated, so this one, this one, this one, this one, this one, and this one, and this one. Okay. The reason that we make a big deal out of unsaturated fatty acids is because they generally contain fewer calories than saturated fatty acids do, so they're better for you in your diet. Another important thing to remember about unsaturated fatty acids is, as you can see, they have kinks in their chain, and what this means is that at room temperature, the more double bonds you have in a fatty acid chain, the more likely it's going to be more like an oil at room temperature rather than a solid like Crisco. Okay, so that's the significance of saturation. If you eat a lot of saturated fatty acids, you're eating a lot of calories because the number of carbon-hydrogen bonds is directly proportional to the number of calories in the molecule. So if you eat a lot of fatty acids, you're eating a lot of calories, your body's going to store that as fat. And we're predisposed to do that because the fat that we store in our body is a way to get us between meals as a calorie source. Fat represents a calorie-dense molecule that takes up very little volume because it doesn't associate with water, and so it's an ideal way to stock our body's pantry so that we can have calorie sources between meals. What element common to amino acids and proteins is not seen here? And the answer is nitrogen. Now, what happens when we take 3 of these molecules and we condense them with a glycerol molecule? And the answer is we form this molecule, which is a triglyceride. Okay? When this molecule was formed from its 4 component molecules, how many water molecules were released? And the answer is 3. Okay? Next question we want to ask, okay, true or false? The 3 arrows indicate sites of dehydration synthesis. The answer is true. Next question. Each of the 3 indicated oxygen atoms is the remnant of the interaction between a hydroxyl and a carboxyl group, and the answer to that is also true. Okay? So what's happening here is just like what I described earlier on in the presentation. The hydroxyl groups, which were attached to these carbon molecules of glycerol, they see the partially positively charged carbon in the carboxylic acid group of the fatty acid, and so they're attracted to it, and this causes them to form a covalent bond and lose the components of a water molecule as a result. And so now we have a process that consumes energy and creates the triglyceride, and this is actually the major fat store, the major fuel store in the human body. It's practical because it's calorie rich and it takes up relatively little volume. If the human body had to store an equivalent amount of calories in the form of carbohydrates or protein, we would be unimaginably large, and this wouldn't be a workable system for the human body. Okay, why is it a lipid? It's a lipid because, again, you have to look at the ratio of hydrophobic components of the molecule to the hydrophilic components. These would be the hydrophilic components up here. These are the hydrophobic components. Well, look how much of this molecule is greasy, okay? So this is going to tend to associate with other greasy molecules because that's, again, going to satisfy the second law of thermodynamics, which is to take on a low energy arrangement if you can do it. Okay, what is this molecule? This is a disaccharide. It's formed from two monosaccharides using dehydration synthesis. And again, the bond that's formed between them is right here. Okay, this is called a glycosidic bond. It forms between two adjacent hydroxyl groups on two adjacent monosaccharides. We lose water as a result. This again is a process that involves an energy input in order to happen. And this is another example of dehydration synthesis. This is maltose, okay? This is a disaccharide maltose. There's two glucose molecules hooked to each other. This is formed when we break down starch or when we break down glycogen, okay? Maltose is generated. Okay, another organic molecule. This is a monosaccharide. This is glucose. Notice again the hydrogen to oxygen ratio, which is 2 to 1. Okay. Notice also in this slide and in the previous slide that these are polar molecules, and the reason for that is they have a lot of electronegative atoms in them. Look at all those oxygens. And so this is going to very easily dissolve in water. And you know this intuitively. You know that a spoonful of sugar will dissolve in water. And the reason for that is that it's very easy for these polar hydroxyl groups to associate with the polar water molecule and go directly into solution. And so you can take a spoonful of sugar, dissolve it in water when you stir, and it'll stay in solution. Now, if you keep adding sugar to this glass of water, eventually you're going to get to a point where you can't dissolve any more sugar in that glass of water. And the reason for that is that when you reach that point, you've occupied all the available water molecules with a sugar molecule. And so the solution is said to be saturated. And at that point, the more sugar you dump in, it's just going to look at you, okay? Chemists call that the solubility product for a particular mixture. It's just a number that indicates how much of something you can dissolve in something else, okay? But if you were to try to dissolve sugar in oil, it wouldn't work. You can even try this at home. Pour you some Wesson oil in a cup and take a teaspoon of sugar and drop it in there, and what you'll see is that the sugar just sits in the bottom of the oil and doesn't dissolve. Now, why is that? Because oil's nonpolar, okay? And the energetically favorable thing for the oil to do is to stay associated with the oil and not interfere with the sugar. And as a result, what you get is a two-phase mixture again, okay? Next question. Which one of the indicated oxygen atoms was involved in the process of dehydration synthesis? And the answer is that one, right? So that's for the reasons that we just discussed. Okay, complete these statements. Glucose plus glucose produces maltose plus water. Glycerol plus 3 free fatty acids produces a triglyceride plus 3 water molecules. ATP plus water produces a hydronium ion plus ADP and inorganic phosphate and energy in the form of both heat and usable energy. The hydronium ion forms because a proton is given off when you split off a phosphate from ATP, and that will associate with the nearest water molecule. An amino acid plus another amino acid forms a dipeptide that contains a single peptide bond, and it also gives off a water molecule. This activity right here, the formation of dipeptides, is carried out by special structures inside living cells called ribosomes. Okay, they are the protein builders. Okay, next question. Which arrow indicates the process of hydrolysis? And that's arrow number 2. Okay, hydrolysis means to use water to split something. Okay, so here water is consumed. Sucrose is a disaccharide made up of glucose and fructose. It's split into its component monomers, and now we can go off and serve our purpose, perhaps as fuel for living tissue. Now, this is kind of an interesting slide here because you can see that you can go in either direction in this chemical reaction, but in point of fact, in living tissue, what happens is that the splitting of disaccharides and the production of disaccharides happens in completely different tissues in the human body and is mediated by completely different enzymes. Remember that enzymes are basically third parties in chemical reactions that help chemical reactions happen because they lower the energy barrier, and they do that by making certain that collisions between reactants are always going to produce a product, and you won't have to rely on changing the concentration or the temperature of the reaction in order to generate enough product to be useful in the time scale that living systems require it. You have to separate these two reactions because if they happened in the same place at the same time, you can see that we wouldn't get anything done, right? We would take glucose and fructose and we would synthesize a sucrose, and then we turn around and do the reverse, okay? And this is what we would call a futile cycle. We would use a lot of energy, we'd generate some heat, but we really wouldn't produce anything useful for the body. Okay, next question. What is this molecule? And it's ATP. This is just the full chemical structure drawn out for you. What are the components of ATP? Well, we've already discussed them. Adenine plus ribose plus 3 phosphate groups. There's phosphate group number 1, phosphate group number 2, and phosphate group number 3. Okay, there's the ribose right there, and there's the nitrogenous base, adenine. Okay. How many high-energy bonds are found in the molecule? The answer is 2. There's one. Let's circle them. I'll circle them in red. Okay. There's one. And there's one. Okay, next question. What color ball was used in the models to represent phosphorus? And the answer is purple. Okay. Moving on. True or false? Water is an organic molecule. False. There's no carbon in it. Steroid molecules are lipids. True, for reasons we've already discussed. Sucrose is a monosaccharide. False. It's a disaccharide formed from fructose and glucose. Question number 4. Glycerol contains 3 hydroxyl groups. The answer to that is true. Next question. Proteins are large polypeptides. Generally speaking, yes. Some proteins are very small, okay, but most are rather large. Next question. Water is released in hydrolytic reactions. False. Hydrolytic reactions consume water in order to split molecules and release energy. Next question. A tripeptide contains 3 peptide bonds, and the answer to that is also false. It contains 2 peptide bonds. Next question. The principal elements common to proteins are carbon, hydrogen, oxygen, nitrogen, and phosphorus. The answer to that is false. The reason for that is phosphorus is not common to proteins as synthesized. Okay. Next question. For a carb and a lipid with similar numbers of carbon atoms, the carb would have proportionally more oxygen The answer to that is true. That's why carbs dissolve easily in water and lipids do not, because they have a lot more oxygen in them. Next question. A peptide bond is a double covalent bond between carbon atoms and oxygen in peptides. The answer to that is false. Okay, a peptide bond is a single covalent bond between a carbonyl carbon and an amino group. We drew an example of the peptide bond earlier. Next question. The carboxyl group